So welcome. Thank you for logging in, everyone. Welcome. This is the session 42 called Integrity of the International IVF Initiative. My name is Dr. Alison Coates, and I'm honoured to be co-hosting this session entitled Integrity for the International IVF Initiative with Dr. Dara Berger. Our continued goal is to provide educational opportunities while bringing our community together. It's great seeing so many reproductive biologists and specialists reaching out from all across the world to participate in these sessions. Thanks so much for logging in, and please ask questions as we go along. I'd first like to introduce my co-host, Dr. Dara Berger. Dr. Berger has been in the field of reproductive medicine since 2009, and she's been an IVF laboratory director since 2014. She's currently the lab director at the University of Pennsylvania's clinical IVF program, Penn Fertility Care, and she's also the off-site lab director at UNC Fertility. Both Penn and UNC have active academic IVF programs, and Dr. Berger currently enjoys teaching REI fellows and medical students. She's also interested in participating in translational research and teaching opportunities.
Thank you for the introduction, Alison. And in return, I will introduce you to our audience as well. Dr. Coates began her clinical embryology career in the United Kingdom in 1987. She received her PhD in genetics from Kent University in the UK under the direction of Professor Darren Griffin. Dr. Coates has over 30 years of experience. She has worked as an embryologist at the Hammersmith Hospital in London, at Leeds General Infirmary, and in the Middle East before relocating to Portland, Oregon in 1999 and created and directed the Reproductive Medicine Laboratory at ORM Fertility. She is board certified as a high complexity lab director and has published many papers and led several RCTs in the field, most noteworthy to determine the best transfer strategy for PGTA screened embryos, which was published in 2017.
Thank you, Dara. So the title of this I3 session refers to the integrity of human sperm. Making sure that the sperm used to fertilize an egg is of the highest quality should be logical and necessary for a successful outcome in IVF cycles. And over the years, we've tried to mimic in vivo conditions of the female tract by filtering out what we believe to be good sperm from the millions of possibilities in any ejaculate by a variety of methods in the IVF lab. We would like to thank Zymot for sponsoring this session. Our two speakers today will compare and discuss sperm selection by microfluidic separation compared to standard sperm separation techniques and the resulting outcomes of IVF cycles. Dara, I'll now turn the session over to you for the speaker introductions.
So our first speaker is Dr. Alessandra Parrella. Alessandra Parrella is a reproductive scientist and clinical embryologist at the Ronald O. Perelman and Claudia Cohen Center for Reproductive Medicine. She holds academic faculty position at Weill Cornell Medical Medicine in New York. One of Alessandra's many projects aims at assessing sperm chromatin fragmentation from different levels of the male genital tract, leading to the utilization of surgically retrieved spermatozoa from these men. Alessandra has been successful in achieving high clinical pregnancy rate by selecting spermatozoa through a microfluidic device, yielding the highest portion of progressively motile cells characterized by the highest genomic integrity. She is currently assessing the gene expression profile of spermatozoal RNA and proteomics in men with idiopathic infertility using next-gen sequencing.
Good afternoon, everyone. I'm going to share my slides now. Perfect. So first of all, I would like to thank the organizers of this meeting. It is a great pleasure to be here and give you a talk on sperm genomic integrity. This is my disclosure. The conventional method to screen for infertility is via semen analysis. Once we consider abstinence, volume, and viscosity, the most important parameters are concentration, morphology, and motility if present. If not, we can assess the viability of the sample. We can also quantify for foreign cells and assess agglutination. But semen analysis does not provide information on genomic profile and embryo developmental competence. While dealing with the type of infertility, it is important to consider that oligo and esospermic men have a higher incidence of chromosomal abnormalities. For example, here in this study from Japan, on about 1,500 men, this incidence is as high as 12%. Indeed, in addition to autosomal defect, there is a large representation of gonosomal abnormalities, mainly related to mosaic or complete form of Klinefelter syndrome. Moreover, these men have a higher incidence of Y chromosome microdeletion that increase when the concentration of the spermatozoa decreases, as you can see in this table. However, once sperm are retrieved, whatever through ejaculation or surgically, they perform quite well in terms of fertilization and clinical pregnancy rate. FISH is the technique that has been used for many years to assess the ploidy status of the spermatozoa. Previously, in 2002, we have investigated the aneuploidy of the ejaculate and surgical retrieved spermatozoa, and we evidence a higher incidence of chromosomal abnormalities in testicular spermatozoa. However, this study was performed on a small population, and we assessed about 100 spermatozoa by only 4 chromosome probes. But in a more recent study, when we assessed the FISH aneuploidy for both ejaculate and testicular spermatozoa within the same individual this time, we found that aneuploidy was higher in the ejaculation, although not significant. In this study, 1,000 spermatozoa were analyzed with 9 chromosome probes. In this manner, sperm aneuploidy was also carried out by next-generation sequencing. When we look at the FISH aneuploidy assessment depicted here in various colors, we can see that it appears minute when compared to the assessment by NGS shown here in green. This dramatic difference demonstrates the sensitivity of the NGS assessment. This is a spermatozoon visualized under transmission electron microscopy. Here you can see a distinct and intact inner and outer acrostomal membrane of the spermatozoon. Beneath the acrosome is localized an important cytosolic factor known as PLCZ. This factor is necessary to activate intracellular calcium oscillation, triggering oocyte activation. PLCZ can be assessed by monofluorescent, and if it is absent, assisted oocyte activation is necessary after ICSI. This is a clear centrosome, which is essential to form the sperm aster during fertilization. The abnormal sperm morphology can give you an idea of the structural abnormalities of the centrosome. Also in this case, we can use immunofluorescent microscopy to assess the presence and the integrity of the centrosome. But why centrosome is important? The centrosome is responsible for the formation of the sperm aster, which guides the migration of the female pronucleus toward the male pronucleus, allowing the juxtaposition of the paternal genome. Now, looking at the sperm organization, it is important to mention that during spermiogenesis, histone proteins are replaced by protamines that allow the supercoiling of the DNA in toroid structure. The rest of the DNA, approximately 16–20%, is histone bound, and these regions are more vulnerable for DNA breaks, increasing the chance of sperm chromatin fragmentation. The histone content can be assessed with aniline blue staining. In this picture, the spermatozoa with the pale blue have a normal content of histones, while those with darker blue have an excess quantity of histones. The tests used to assess the DNA damage are Tunnel, SCSA, and Comet. SCSA and Comet are able to detect single and double DNA breaks, while DNA assess the overall DNA fragmentation. Previously, we investigated the relationship between sperm chromatin fragmentation and paternal age, and we saw a concurrent increase between paternal age and sperm chromatin fragmentation. In addition, we also wanted to see if sperm chromatin fragmentation is related to the days of abstinence. And if we rank the patients with abnormal sperm chromatin fragmentation according to the length of abstinence, we can see that the percentage of the spermatozoa with DNA damage is higher when the days of abstinence increase. Sperm DNA fragmentation can predict the outcome for the couple undergoing intercourse, programmed intercourse, or standard in vitro insemination, but almost never with ICSI. It has been proposed by Simon and Lewis that ICSI performed well even with DNA fragmented spermatozoa. This because spermatozoa are not exposed to culture media overnight, and also the oocytes are not exposed to reactive oxidative species released by spermatozoa. Nevertheless, sperm chromatin fragmentation does affect embryo implantation, which can lead to miscarriage. We have previously described a treatment algorithm that guides the reproductive intervention of couples who failed to achieve a pregnancy after 3 attempts of IUI. I have to mention that this study was conducted with a young woman and normal semen parameters. So the men are offered to undergo a sperm chromatin fragmentation assay. That, if it's normal, they are treated with standard in vitro insemination. Those with abnormal sperm chromatin fragmentation or who had failed to achieve a pregnancy with standard in vitro fertilization, we offer ICSI, which is able to achieve a pregnancy rate of 18.7%. Now, the question is how we can treat couples who fail even with ICSI. And we found that these men had extremely elevated levels of sperm chromatin fragmentation in their ejaculate. We have learned that there is an increase in sperm chromatin fragmentation as the specimen progress through the male reproductive tract. In a previous study, our team, alongside our reproductive urologist, wanted to assess the sperm chromatin integrity throughout the male genital tract. Here is the complete mapping of the male genital tract with the associated sperm chromatin fragmentation levels. The 83 men who consented to be included in the study had severe DNA fragmentation in their ejaculate, reporting 32.9% with tunnel, which fell to 20% in the vasa deferens on average, a little bit less than 16% in the epididymis, and just 11.4% in the testis. If we compare the overall clinical outcomes between ejaculate and surgical retrieved, we can see that although the fertilization is slightly lower in the cycle with surgical retrieval, the implantation, clinical pregnancy, and delivery increase. However, not all patients are willing to take the risk associated with surgery. In our center, we perform almost 90% of ICSI cycle. About 10% of these cycles are performed with surgically retrieved spermatozoa. From a study accomplished in 2014, we have learned that there is a strong inverse correlation between sperm motility and chromatin fragmentation. Therefore, when motility increases, DNA fragmentation decreases. Based on this, we have studied a new method known as microfluidics to select spermatozoa with the highest motility and the lowest DNA damage. There are 3 Zymote devices, and in our laboratory we are using the 850 microliter that is called the Zymote Multi. This is how it works. As you can see in this animation, only the spermatozoa with DNA integrity and progressive motility are able to reach the upper chamber. And we presented our preliminary result last year in Journal of Assisted Reproduction and Genetics. Our aim was to select spermatozoa with the higher chromatin integrity from the ejaculate spermatozoa. To do this, we divided the semen sample in 3 aliquots: the raw sample, the specimen processed by density gradient, and the specimen processed by microfluidic sperm selection. We assessed the sperm chromatin fragmentation on all 3 specimens and compared the result. We used tunnel assay to evaluate the fragmentation in the spermatozoa, with the normal threshold being less than 15%. Tunnel tests can give you an idea of the DNA damage in spermatozoa, but is unable to distinguish between single and double-stranded DNA breaks. In this paper, Casanova et al. Evidenced that double-stranded DNA breaks can cause delay in embryo development and can impair also implantation. Therefore, in our study, we also evaluated the double-stranded breaks in the spermatozoa using a neutral comet assay with the normal threshold being less than 3%. For this study, we involved 69 men. The motility improved after density gradient from 33.6% to almost 60%, reaching 97% after microfluidics. At the expenses of the concentration, the morphology also improved. Here I would like to show you the motility of the raw sample, density gradient sample, and the new selected spermatozoa method, microfluidics. But very important, we had a remarkable improvement in sperm chromatin integrity. Indeed, after density gradient, sperm chromatin fragmentation was above our threshold. I just would like to remind you that our threshold for tunnel is 15%. In this case, it was 18%. But after microfluidics, it was just 1.5%. And this improvement in sperm chromatin integrity was shown also in the previous I3 section by Alicia. The double-stranded DNA breaks also decreased from 4% to 3.3% after density gradient, reaching only 0.3% after microfluidics. Confident in this improvement in sperm chromatin fragmentation, we then proceed to use microfluidics in couple with history of ART failure and high DNA fragmentation. The first group that I will describe includes a couple who fail ICSI with density gradient and were treated by microfluidics in a subsequent cycle with a fresh embryo transfer. This group includes 11 couples. In this table are reported the semen characteristics after density gradient and microfluidics. Sperm chromatin fragmentation remained abnormal after density gradient, while after microfluidics decreased dramatically. Here we have the patient's demographics, in particular the maternal and paternal age. This couple underwent 22 ICSI cycles with density gradient and 12 ICSI cycles with microfluidics. Fertilization and embryo transferred were somewhat comparable. And when we look at the clinical outcome, we can see that with density graded, the implantation rate was only 5.2%, with 13.6% of clinical pregnancy rate and very high pregnancy loss. After microfluidics, we have an implantation of 33.3% with 66.6% of clinical pregnancy rate, and in this group we didn't have any pregnancy loss. The second group that I'm going to describe includes couples who were treated solely with microfluidics and underwent fresh embryo transfer. This group includes 28 couples. After microfluidics, sperm chromatin fragmentation decreased from 22% to 3.3%. This is the maternal and paternal age of the couples. A total of 35 cycles were performed, achieving a fertilization rate of 76.9%. Looking at the clinical outcome, we were able to yield an implantation rate of 25.3%, 48.8% of clinical pregnancy rate that resulted in 39.3% of delivered. Pregnancy loss was 18.7%. The last group includes couples who were treated by microfluidics due to history of high embryo aneuploidy with densely gradient sperm selection. In these 29 men, besides the enhancement of motility and morphology after microfluidics, also the chromatin integrity improved. This couple underwent 42 ICSI cycles with density gradient and 35 with microfluidics. The fertilization rate was higher in microfluidics cycle. 109 embryos were screened for PGTA after density gradient and 184 embryos after microfluidics. Interesting, the number of normal embryo obtained was much higher in the cycle where microfluidics was used. Indeed, in cycle with density gradient, the embryo aneuploidy rate was only 23.8%, while in the cycle with microfluidics was 48.9%. While the cycle treated by density gradient failed to yield any pregnancies, those with microfluidics had 65.5% of implantation with 73% of clinical pregnancy rate and very minute percent of pregnancy loss. In conclusion, in this study, we confirmed that sperm chromatin fragmentation is strongly correlated with sperm motility. Microfluidic yields the highest portion of motile spermatozoa with the least DNA damage. Spermatozoa with intact DNA improve clinical outcome. Minimizing the double-strand DNA breaks enhance euploidy embryos. I would like to thank all my colleagues that contribute to this study, in particular my colleague, the ICSI and andrology team, and my mentor, Dr. Giampiero Palermo and Dr. Zev Rosemax. Thank you so much for your attention.
Thank you so much for your talk. We have one question for you before we move on to Dr. Anderson, and it's from our audience. Have you compared the Zymote processed sperm with other swim-based separation techniques, like having sperm swim from raw semen into a drop of media under oil?
No, we didn't compare. We did, as I shown, we just compare specimen processed by density gradient and microfluidics.
Okay, thank you.
Thank you.
Okay, so our next talk is by Dr. Tony Anderson. Dr. Anderson completed his Master's in Clinical Embryology Studies at Leeds University in Leeds, England, under the direction of Professor Alan Handyside and Dr. Helen Picton. Tony's studies focused on cryopreservation and non-invasive methods of predicting euploidy in early embryos. Tony completed his doctoral studies at Nova Southeastern University, a private university in South Florida. His studies included comparison of cryopreservation methods of oocytes and pre-implantation genetic screening of embryos for optimized outcomes. Thank you, Tony.
Thank you, Alison, and very good presentation, Alessandra. That was very, very, very well done. I would also like to thank Zymote for the for the invitation to come speak today, as well as the organizers of I3. I'm delighted to be here. Today I'm going to talk about euploidy rates and pregnancy outcomes using microfluidic sperm separation, particularly the Zymote device. A lot of this is based on some of my experiences with what I presented at ASRM this year, and then breaking that down some. These are my disclosures. I'm on the scientific advisory board for INVO Bioscience. And the title of this session is integrity. So today I want to talk about the integrity of the sperm quality, integrity of the clinical outcomes, the chain of custody and the workflow. We'll get into some of that, going a little bit of a different direction, and the ease of use of the device, and demonstrate that for everybody today. So when we first started, when I first got into human IVF, the standard protocol was doing a raw semen swim up. We would just put raw semen in the bottom of the tube, put media on top of that, allow it to swim up. This is pre-ICSI days. And so, a lot of the male factors we had, we just simply did not get enough sperm, and density gradient changed our world with we're able to harvest more sperm, clean it up, and get better sperm. We used to just order the per call, what it was called, through Sigma or other chemical vendors. And it was really just an ideal solution for filtering out biological materials. And it's really just PVP coated silica beads. With the FDA requiring approvals, they came out in other versions called Sperm Grad or Isolate, all very good products. So, going forward, the last 30 years of my career, I've been using the gradient separation methods, as most of us have. Most of the people in my laboratory have never even done just a raw semen swim up like we used to do in the early 90s. Gradient has just been the standard protocol, and a lot of people use gradient for IUI and IVF preps. And so I decided I would try the Zymote device just to see what it would do, and just see what kind of clinical benefits I might get out of it. The whole goal of it is it's a very small pore, 8 micron pore in the membrane that the sperm can swim through, very much like the raw semen swim up we used to do pre-gradient. The goal of this is that there is no centrifugation when you're done. There's no concentration of the sperm, and nearly 100% of the sperm are modal, as you saw in Alessandra's presentation. So in this study, we use our density gradient protocol. I just use a single layer density gradient, spinning it for 10 minutes at 300 Gs. I use a HEPES buffered wash media, sperm wash media, spin it for another 10 minutes at 300 Gs. And then I dilute it out to less than 10 million per mil and do ICSI from there. With the Zymote, I have only used the 850 microliter device. It's a real simple process of loading the sperm, loading the media, incubate it for 30 minutes, and then do your final count, and then do ICSI with the 2 samples. This is a retrospective study. Of the data. We looked at 2 time periods with the density gradient, and then a time period with the Zymote device. All the oocytes were inseminated with ICSI under low oxygen incubation. All the embryos for PGTA, all the data in this study were PGTA patients. Were laser assisted hatch on day 3. All embryos were biopsied in day 5 and day 6. There's no day 7 data included here. Away from the, of course, away from the intercell mass. And all patients had greater than 1 million total modal sperm to be included and be used for Zymote. This is the data I presented at ASRM. When I first started looking at the data, my goals originally were just to see what clinical improvements we would get. I used it for the first 50 patients and realized the workflow patterns were were very kind, and so and that it didn't make things any worse. And so went on for another year looking using the device, and this is what we originally noticed, is that we started getting more euploid embryos, particularly in the day 5 patients, very similar to what Alessandra presented as well. There was no difference in the day 6 embryos, euploidy, and there really no difference in implantation rates and ongoing rates between the two. A lot of the questions I had from ASRM were out into different age groups. I have a little more data. I'm going to show that here. These are between the Zymote and the gradient. There was, with the patients less than 35, less than or equal to 35, there was no difference in fertilization rates, euploidy rates, almost exactly equal numbers of euploidy between day 5 and day 6. Positive betas and the ongoing pregnancy rates equal in numbers. Interestingly, we had a higher rate of miscarriages in the gradient group, and I would probably say that is primarily due to actually being able to have better quality sperm with lower DNA fragmentation, virtually no DNA fragmentation. Where things get interesting, and I think that it's important to note here, this is the important group where the patients greater than 35. We start to see a difference in euploidy between the day 5 and the day 6, between the Zymote and the gradient. And although we did have significant higher rates of euploidy with day 5 embryos, we did not reach significance with the day 6 embryos, but the p-value on this is 0.09. So it's getting pretty close to significant, the Zymote, with the Zymote device. Once we have a euploid embryo, it doesn't surprise me that we have great positive pregnancies and great ongoing pregnancy rates. And there was no difference in pregnancy loss rates in the Zymote versus the gradient group here. In this video, I'm going to demonstrate the gradient on the left-hand side and the Zymote on the right-hand side. Everybody is very familiar with using a gradient process. I actually, we use actually colored tapes to differentiate between patients. We'll have their patient name and ID numbers and accession numbers on there. You can see up here in the top left corner the loading of the device with 850 microliters of the raw semen. I'm going to pull out some of my sperm wash media, which is just basically Heapy's buffered media with 10% HSA in it. Pull up 750 microliters in a syringe and drop that over the top of the membrane. You can see here in the top right-hand corner here the dropping of that media. You want to fill that little chamber on the side, and just basically just distribute that 750 microliters over the top of the membrane, and then you're just going to stick it in the incubator for 30 minutes. On the density gradient, we're all familiar with that. I personally do a single wash with my gradient, and then if some people will do 2 for time purposes. I only demonstrate here with the 1 wash. You can see on the right-hand side the timer going off, timer counting down. It's 30 minutes. Whether we're using a density gradient or Zymote, the timing of this is about the same time. So in our, this wasn't part of the original study. We didn't do the DNA fragmentation on our patients that were in the study, but I've done some DNA fragmentation with a raw ejaculate and post-zymote since then. And this is what it looks like using the halo sperm assay. You can see the bigger the halo, the better. And you can see on the left-hand side, the sperm that don't have the halo in the raw ejaculate, and the sperm on the right-hand side. I actually didn't find any sperm that were DNA fragmented. I think Alessandra showed with a tunnel assay of 0.3%. I've looked at 10 patients in my study I'm working on now, and it ranges anywhere from 2 to 50% DNA fragmentation with an average of around 19%, but virtually 0% post zymote prep, which I know a lot of other data has said that. And, you know, and I just wanted to kind of prove that for myself, show that for myself. So one of the final things I want to talk about here is the takt time and in the process management of things. In Six Sigma, I know there's been a couple talks at ASRM about this year as well, in the process management in the laboratory. Toyota in the 1950s went through a process of lean management in their car manufacturing. We're starting to use some of those same technologies in other areas of industry. And one of the things I really like about I talked about the workflow. And the takt time is basically, it's a German word from Taktzeit, meaning cycle time. And the Poca Yoki is error proofing. And everything we do in fertility is all about error proofing what we do, but we don't give a lot of work towards the cycle time. And one of the things I've really looked at in the andrology lab is I could process a sample. I could put on a sample, and 10 to 15 minutes later, put on another sample and another sample, and another sample. And if you look at the 6 samples that a sample would take for 30 minutes, if you're doing them individually, it's 180 minutes, about 3 hours worth of time. But I could technically, if I did this, I could do all 6 samples within 1 hour, saving you know, personnel time and you know, the FTEs that we required in the lab. If I look at it with the gradient process, part of the takt time process is where you get bottlenecks between different times for each procedure. Being with the zymote, you're basically putting the sperm on and taking it off. There's only 2 steps. With the gradient, you have, if you're only doing 1 spin or 2 spins with your washing the gradient out, it's going to you know, get a bottleneck, and ultimately you can only process 1 sample at a time. So you're going to still need that, you know, 3 hours to process 6 samples. So for me, when I first started this, it wasn't so much even about what I was going to get out of clinical outcomes. So I'm glad I am seeing some improvements in my clinical outcomes. But just the workflow ease for me was worth continuing on with this device. The added value of the improved clinical outcomes, I'll keep using it for all my IVF patients. So I'm going to kind of wrap things up here, and you know, just kind of final, you know, just reemphasize the fact that we do get more euploid embryos with the zymote, and ultimately in those cases, especially the patients that are greater than 35, you know, those are where we really need the euploidy, and so we need to get as many embryos we can get that can ultimately turn into babies. So more euploid equals more embryos over the cumulative rate of in our transfers. There is we did have a higher reduction in losses in the patients that are less than 35, and just, you know, simply an easier method to use, more use of your resources. And the whole goal of this is in the Poca Yoki, you know, is the just error proofing of it, and, you know, safer system for where mix-ups could occur when you have to go from tube to tube. What I like about this study, and I actually enjoyed Alessandra's presentation as well, I think the PGTA is a great control for this because we can see what is normal with the euploidy, and when we transfer those, what actually turns into a pregnancy. And finally, I get a lot of questions of what could we use this for? Why do I use it for IUIs? I have not used it for IUI, and it's not so much that I don't. I think it would make any would make it improvements. I already have I just do a straight wash on my IUIs, and so it's already a two-step process. I basically wash the seminal fluids out, and I'm done. It's basically a 10-minute procedure. So that's really the main reason why I haven't used it for my IUIs, although I do know that there are some studies showing some improvements with this. One of the things I do like about the device is that if you didn't have a laboratory team to do IUIs with, the whole device is set up. You can just pull off 0.5 ml and be ready for your IUI immediately following that. So thank you so much. Sorry for the technical glitch there.
Thanks, Dr. Anderson. That was a great talk. Another question from the audience is, have you tried it with standard insemination, so not ICSI, with the device? Have you used it for regular insemination in IVF?
I have used it for my INVO cells. I do all ICSI on my IVF cases. I have used it on INVO cells. I've had mixed results with that. With INVO cell, you don't get to see fertilization, and so you don't know if they fertilized. I've talked to DX now about other people using it with conventional INSEM, so it's a really good question. And I do know that the people that are using it with conventional INSEM will wash it after the zymote, and I'm not really sure why, if that makes things any better, but most of the time when I'm using it for my IVF cases. I had mixed results with my INVO cases, so I have gone back to using density gradient on my INVO cell cases.
I have a question, and I think this is for Alessandra. What is the reason you believe there is a big difference in euploidy? There have been a couple questions from our audience members. They're curious if it is due to the device and what the mechanism would be to yield more euploid embryos.
This is a good question and not easy to answer. So what we saw is, of course, a decrease in DNA fragmentation and also in double-stranded DNA. So we think probably that since the sperm is more integral, so we don't have any breaks. Probably also the oocytes mechanism repair can probably repair better the few gaps that there are in the spermatozoa. So decreasing the sperm chromatin fragmentation probably could help better also the oocytes to interact with sperm, with the DNA of the spermatozoa.
Tony, do you want to add to that?
Well, it's when I first started this, I actually didn't expect there to be more euploidy. It was one of those kind of aha moments. I was really hoping to see lower loss rates because one of the things that drives me crazy in this business is why do we have loss rates after we transfer a perfectly normal euploid embryo? And so originally, when I just looked at the data just overall, it did not see that. But then when I split the data out, I did see some differences. I really think it's because we're just, as Alessandra was showing that, you get better sperm and you're ultimately getting a better embryo that's available to implant. And, we're also seeing possibly lower aneuploidy in that sperm. We never really think aneuploidy is coming from the sperm, but it possibly is adding more to it than we know.
Okay, and then a second question is from Eunice Dodd. How is the efficacy of Zymot in low concentrations of sperm?
I can answer. So I used a sample with very low concentration and also low motility. I have to mention that there is another microfluidics. So I use the 850 microliters, but there is another chamber that is called Zymote XC, which that you can use also with low concentration. So we have 3 devices, and the Zymote XC is very good for the oligospermic patients. Besides that, I also worked with 850 microliters with oligospermic patients, and also with very low motility. And although the concentration decrease, so probably you have to search a little bit in XC, but still the quality is very good. So we still prefer to proceed also with oligospermic patients.
So I think, could you give a number, like what is the lowest concentration that you had success with?
Yes, so I depends also from other parameters, because I think it's very important also the motility. If you have low concentration but you have good motility, you still have very high chance to have spermatozoa in the upper chamber. So I have worked also with the low, very low. We can say even with 5 sperm per macler, 0.1 million, so very low. But again, motility is very important in this case. Yeah.
I have a question for both of you actually about your studies. And did you think about doing maybe sibling oocyte studies at all to compare in the same cycle with both methods, just to compare the euploidy rates with density gradient and the Zymote?
So we personally didn't do it, but yes, so I think it's a really good idea. And actually my idea is also to use donor egg to see the impact of the spermatozoa. So far we didn't do, but of course there is a lot that we would like to try. So, but thank you for this suggestion.
Tony, did you do any sibling oocyte studies?
Can you say that again, Alison?
So sibling oocyte studies, they're kind of like your ultimate control, really, right? Did you do any? No.
I did not do that here. That's a great idea. I started this just to see what my clinical outcomes would be, and then turned into a retrospective analysis of the data. I do like the idea with donor eggs, taking the genetic piece out of it. I think we would also probably want to try to do fresh donor eggs, because you don't know if the cryo preservation process is interfering with that. But that would be a great control. I want to kind of talk about some of the the low concentrations. I mentioned we had to have greater than 1 million total modal. But one of the questions I'm always getting from my andrology lab is, when you have a 5 mil sample and you're only using 850 microliters of it, you might have 5 million total modal, but by the time you take your 800 microliters, you're not getting that whole 1 million total. And so I think we just probably just need to work out what works. I have put some samples on, and I've left them on longer, hoping to get some sperm, not with great success. So in our lab, if there's 1 million total modal, the goal is to use Zymote, particularly after seeing that we're getting some of these better clinical outcomes. I really want to try to use Zymote as much as I can. My lab got scared a little bit and started going to the density gradient because, I don't have enough sperm. And I had to show them that, yes, you do. I do think we probably need to do some more studies to see what that cutoff is, and just go from there.
What about biopsy samples?
I don't think your concentration's enough for that. You might be able to use, I haven't used the smaller device. Maybe Alessandra could speak to that. I haven't used the little small chambers. I would love to try that, especially with some of our really low sperm counts. I talked to DX now about the larger chambers to add more volume of sperm to it. But with that, you also have larger volume of media in the top of it. So you have like, you might be able to put 3 mils into the chamber, but you also have, 3 mils of media. And so you'd have to spin that down. And I don't know if that's a bad thing. I actually have done some studies with my DNA fragmentation where I look at the DNA frag after the spinning, and I don't see, immediate increases in DNA fragmentation. Just my initial survey so far.
So Alessandra, did you have any experience using testicular sperm with the small chambers, the tiny chambers?
So I actually just did once with epididymis sample. In that case, we split. So we did alpha. We inject alpha oocytes with epi and alpha with normal. Without processing the epi with the zymote. We had the same result, same quality, same number of egg, actually of embryo that reached the blastocyst stage. So we didn't have any difference with epididymis, but I just did one patient, so I don't think that we can have any conclusion on that. It worked, yes, it worked because the epi was good motility, had good motility, so it was fine.
Thank you. There's a question here from, sorry, Dara.
It's okay. You can keep going.
Okay. So Bill Vanier asked if these were all fresh sperm samples. Any data on frozen thawed sperm?
I personally have used it with frozen sperm, particularly donor sperm. If I have donor sperm, a lot of the samples come with 0.5 ml. I just, add 350 microliters of media to that and put it on the device and have great results with it. So I don't, if you have the numbers of motile, I think the frozen thawed sperm would be great. And interestingly, we know that the freezing process increases the DNA fragmentation. Some of these studies I'm looking at with my frozen sperm shows that as well with the halo sperm. And I have not done zymote or DNA frag on those cases afterward, but the cryopreservation does cause DNA fragmentation. So the zymote's going to make that better sperm when you're done.
Mariana from Portugal wants to know, what is the difference between MFSS and normal swim up results?
So, sorry. Personally, I don't have this data because we usually use, in our laboratory, andrology laboratory, we use density gradient and microfluidic, so I don't have much experience on swim up. So I don't know, but I think there are some studies in the literature where they say that sperm chromatid fragmentation, although is lower after swim up, after microfluidics is much lower. So I think this can help us to understand that after microfluidics, the spermatozoa is much healthier in terms of sperm chromatid fragmentation.
Okay, and has anyone looked at the impact of male age on the pregnancy outcome and using Zymote?
I did not look at the male age. Basically our patients are, looked at the female age less than or equal to 35 or greater than 35, but I did not look at the male age in this.
Do you think you would see a difference?
Me personally, I don't. No, I think if you have enough sperm, by the time you get it through the device, you're going to have good sperm when you're done. And if you don't have enough sperm, it probably is not very good quality sperm. And with you're doing with ICSI, it's not about the quantity that you get; it's really about the quality that you get. Maybe with IUI, it may have an impact, but I couldn't speak to that really.
So if I can add something, in our study, we included patients with advanced paternal age. So as we know, paternal age is related to high DNA fragmentation, and this was the reason why we included these patients, because they presented high DNA fragmentation in their spermatozoa. We processed with microfluidics, and the sperm DNA fragmentation was much lower. We had an improvement, and we also got pregnancy after after microfluidics ICSI cycle.
I have a question for probably Tony, I think. You do your PGTA on quite a few embryos, presumably, quite a few cases of PGTA a year. Did you look at mosaic rates in the results from the density gradient versus the Zymot?
I did. I just took a quick glance at it. Our mosaic rates, I think, are pretty much what we're seeing across the industry, around 10% between low-level, high-level mosaics. I did not see a difference between the two. I didn't include that here. But I don't know if, I've always, that's a whole nother presentation with, I've always thought maybe biopsy method might be causing the mosaicism. My personal opinion, I think most embryos are mosaic. It's just, what level of mosaic are they if they become euploid? I don't think Zymot's going to play an impact in that personally.
Thank you.
Okay. Hussam from Egypt wants to know if any comparing Pixie or Max with Zymot has been done.
I haven't compared Pixie to Zymot. I have done a lot of Pixie. I was actually part of the FDA trials for the Pixie dish years ago. It was a multi-center trial, and I believe, one of the things with the FDA is that it didn't matter if it made things better. Just did it make sure that it didn't cause any harm? And and it didn't. Through the multi-center trial, we did see an increased, pregnancy rate, fertilization rates. In my lab, when I was in North Carolina, we did not actually see any really great improvement from that. I have done some Pixie, and every now and then a patient will request Pixie. If I had to choose between Pixie and the Zymot today, I'd probably go with Zymot because I know that I'm getting good quality sperm on the other side of that.
Has there been any cost analysis done using these devices routinely?
We didn't do any. We didn't do this analysis, but I'm sure that the costs are much lower compared if we have to perform another ICSI cycle, because probably the first one with other semen preparation didn't go well. So that's what I think.
Yeah. The cost of the devices is going to be higher. You're using, I haven't done an actual cost analysis breakdown, but if you were to, using less tubes, you're using less media. One of my biggest things was, aside from cost, was the fact, and that's why I included it here, was the workflow patterns in the laboratory. To me, that far outweighed the cost, and is the benefit that I got from the safety, because it's basically one on, one off system, and just the workflow pattern in the lab. Like I could actually do multitask. If I only had 1 or 2 samples to do, I could be running endocrine machine as well as the andrology side. They're both in the same lab. So you can multitask and do more with that. So for me, it was really about the cost, the increase in cost for the savings in time far outweighed the cost of the device. I do know some people within the Prelude network that are they're charging a little more. You can actually spread that cost out across all patients, just increase your cost of your ICSI or something like that a little across the board for everybody, $100 or something like that. And now it's a break even, and that's in that process. I don't believe there is a CPT code for Zymo yet. And, there is a CPT code for gradient separation. At some point, maybe there will be, and insurance will pay for that. But right now, it doesn't.
Thank you. I think we're down to, I'm sorry. We're down to our last two questions, and I'm going to let Alison ask those.
Sure. Well, Mariana from Portugal asked about the high temperature affecting sperm DNA when you incubate sperm. So she asked, have you tried to incubate Zymo at room temperature and compare the results? Either of you.
I personally have not. I follow their protocol to the letter, 850, 750 media, 30 minutes in the incubator, 37 degrees. I would love to try that. And the reason why I haven't is because they are a little bit expensive. I don't want to waste them. I do actually have in my training lab where I train embryologists, I actually have some devices, and just got these. I'm anxious to try some of these things you're talking about, swim up versus Zymo, different temperatures. Could you put it on a stage warmer? Do you need the incubator? Because that is an extra thing we may not have in the lab.
Mm-hmm.
How about you, Alessandra? Have you tried different temperatures?
No, we didn't try. We always follow the protocol of the Zymo, and now we just incubated 37 degrees. Sometimes actually what I do is I work on the time. So if I have a good sample, especially for ICSI, if I have a good sample with a high concentration, instead of spend 30 time, I just do like 15 minutes or 20 minutes. That's probably I can get probably the best sperm that reach the upper chamber. So this I work more on the time rather than the temperature, but yeah, we can try.
Yeah, I haven't gone less time. I have tried and gone a little more time with the lower sperm concentrations. Not with a lot of luck, but I'll have to try that less time. That's interesting.
Yeah.
And then the last question for today, there's a question about automation in the IVF lab. We're always thinking about ways of maybe automating certain processes. Is this one of the steps you think that could be probably automated in the future? Is it the next step towards a lab on a chip?
My personal opinion is yes. Yeah, just like you could pull up, serum sample, put it into a test tube, and get a E2 value in 15 minutes. It certainly is doable to, put this on a machine, and it just does it for you. One of my things with the machines is, they're not cheap. Just like any hormone machine is not cheap to have or operate or quality control. So, much like CASA systems, not many of us have CASA systems in our laboratories because they're not cheap. Most of the physicians would rather pay a human body to do that. So that's going to be the thing we're going to come against when it comes to the automation piece of it.
Thank you so much, both of you, for a great talk and answering all of our questions. For those of you who are watching, it's live because I'm sitting here fighting with the sun. So thank you everybody for taking the time to participate in this session and for this great discussion. For videos of previous sessions, please go to ivfmeeting.com. You will also find information about upcoming sessions on that same page. I would particularly like to thank the two speakers, Alessandra and Tony. Also, I would like to thank the organizing committee of this initiative, Jacques Cohen, Peter Naj, Thomas Elliott, Shaista Sujuran, Giles Palmer, Marianne Sevetes, and for today's session, Liesl Nell Tamat was a huge help to Marianne, Giles, and Peter triaging questions for the audience. For those of you watching, you may not realize there is so much work that goes on behind the scenes to pull this off. I know I have been a participant on many webinars during these COVID times, but this I3 organization has really mastered the process and has consistently selected the highest quality guest speakers, this session included. This session and the presentations will be available on a new website very soon. Thank you, Thomas Elliott, for arranging all of this. And lastly, our next session will be next Tuesday, December 1st, same time depending on your time zone. It's sponsored by Hamilton Thorne and will be titled Back to the Basics with a Twist, a session looking at variation in our modern lab protocols such as oocyte biomarkers, day 7 embryo transfers, and embryo biopsy from vitrified and warmed embryos. For those of you who will be celebrating Thanksgiving this week, I wish you a safe and happy holiday.